So how do people predict which genes are going to be orthologs? So we have to use a similarity-based methods to do this. We take every pair of genes and we measure their similarity. The most common way of doing this is with BLAST. And then after that, we want to cluster the most similar genes together because we assume that more similar genes will be more closely related. So methods for that include Markov clustering, hidden Markov models, and cliques. Cliques are kind of a graph theory idea where a gene cannot be included in an orthologous group unless it matches with all of the other genes in that group. So these methods aren't without their problems, they have challenges. Sometimes you'll get orthologous groups of genes that are too inclusive. If you allow genes into the group that aren't so similar to each other, you might get paralogs in there mixed in with the orthologs. So in green here, we have an ortholog and then one of its paralogs, so duplications from species number 3. And you cannot use paralogs to build species trees. They will confuse your phylogenies. So the other problem you can get is that the group could be too exclusive. If you restrict the kind of genes that you can let into the group, you might be missing orthology from distantly related species because maybe this gene just evolved and changed a lot over time and now is unrecognizable even though it really was an ortholog. And you can get these two problems in the same analysis. It's not like one method is always going to be too inclusive or too exclusive. So it can be very complex, and people have addressed this in many different ways. Here's a little short synopsis of some prediction programs that people have made in the past. There was OrthoMCL in 2003. It uses BLAST and Markov clustering. Then there was OMA in 2008. It uses Smith-Waterman and then cliques for clustering. Then there was Hansen in 2009. It uses BLAST-P, just like OrthoMCL, but then hidden Markov models for clustering. And then OrthoFinder is almost the same as OrthoMCL, heavily based on it, but uses adjusted BLAST-P scores to factor in the bias of sequence lengths. So I wanted to use these 4 orthology prediction programs to address some questions about orthology prediction in general. So I wanted to know, do the program runtimes scale well if we add more taxa? As we sequence more and more organisms, will these programs be able to handle it, or will they just take forever? How different are the groups that these programs produce? Do we see any meaningful differences there? I would expect that a lot of the times they should predict the same thing, but that every program will have its own strengths and weaknesses and find some groups of genes that other programs miss. And then if we use these groups to make species trees, do we also see differences there? Does it matter in the long run, or do you get the same trees no matter what? So for my methods, I ran these 4 programs on the same data set for this presentation. That is a bilaterian transcriptome data set, so mollusks, annelids, brachiopods. And then for each of these programs, I subset the data into smaller random groups and measured the run times for those. Then I looked at the groups to see if they were coming up with the same groups or with different groups. And then I made species trees from each of these different sets of groups and compared the trees using the Robinson-Folds distance metric. So here are my results. The y-axis here is the different run times. So these programs took between 1 hour and 12 hours to run, depending on how big the data set you use is. When you use only 5 taxa, all the programs pretty much take the same amount of time. But as you add more taxa, some of them start to slow down. OMA is really slow, and it takes longer and longer. It's probably because it's trying to calculate the cliques for all of the genes, which should take quadratic time, is what we would expect for that. But then OrthoMCL and OrthoFinder have close to linear time, and they're faster, which means that we can expect that they'll still be fast even when we add more species. So that's good news. Now, if we look at the entire data set, what kind of groups are we getting? So what this graph shows is we have OrthoMCL, and on the x-axis we have number of taxa represented in the orthologous groups. So what this means is because the peak is around 15, that means that for OrthoMCL, most of the groups that it makes have at least 15 taxa represented, so 15 sequences. If you wanted to have a more complete matrix for your phylogeny, you would want more taxa in each group. Then there's hamster, similar distribution. OrthoFinder, similar distribution. OMA has a really different distribution of groups, though. Most of its groups it finds are less than 10 taxa. So do these programs agree with each other? Are they coming up with any of the same groups? So I found out not as often as you would think. Here are all of the groups found by OrthoMCL, by OMA, Hamster, and OrthoFinder. So there is some overlap. Also, this isn't to scale, so I'm going to put some words up here to kind of talk through it a bit. OrthoMCL and OrthoFinder agree most of the time. They find hundreds of genes, hundreds of groups of genes that are all the same. So that makes sense. They actually use really similar methods. That red area should be much larger than it is because 98% of OMA groups were only found by the OMA program. That's because they were much smaller and the other programs were either ignoring those groups altogether or maybe combining them into larger groups. And the overlap between all of these programs was very small. There were only 4 orthologous groups of genes that were found by all of the programs. So we know that these programs are producing different output, but does it affect the species trees? So when you change the methods that you use to make your species tree, you will often get different trees. For example, if you change the optimality criterion of, say, from maximum likelihood to maximum parsimony, then you will get different trees. And so that's what I did here. And when you change the optimality criterion, you get 2 different trees, and you can measure their distance using Robinson folds. So what I wanted to know is, if we do this for changing the orthologous groups that are used, do we see a comparable difference? Do we see greater differences? Or do we see a negligible difference in the trees? So if you only change the optimality criterion but leave the input data the same, I got differences between 8 and 10. But when I did this with changing the orthologous groups, I got much higher distances. I'm going to highlight all of the times when the difference is higher. And it's not all the time, but more often than not, you get more different trees by changing the orthologous groups than by changing the optimality criterion. So to recap the results, OrthoFinder and OrthoMCL were the 2 fastest programs, and if you have a large data set, you can expect them to still run quickly. There was far less overlap between the predicted groups than expected. Only 4 orthologous groups of genes were predicted by all the programs. And the orthologous groups had at least as big or bigger impact on the final species tree as the choice of optimality criterion. So for future work that I want to do, I want to add more orthology prediction methods. I listed a couple here, but there are many more. And I also want to consider Bayesian phylogenetic inference. Doing both of those things will allow me to have more final trees to compare and really allow me to say something statistically meaningful about the difference in the trees. And then I also want to do this with some more data sets independently, completely different sets of organisms to see how widely applicable this is. And that's something I'll be doing this summer. And I think that this preliminary work shows that orthology prediction does matter and makes a good case for developing new and improved orthology prediction methods in the future. So that's the end of my presentation. I would like to thank the Richard Gilder Graduate School at the American Museum of Natural History, all of my classmates and professors from there, my committee, the Wheeler Lab, the Invertebrate Zoology Department for making this research possible. And thank you for listening, and I think I have time for questions.